So with that, our next presenter is Dr. Phillip Lujan. And so Dr. Lujan and I are in the same department at NMSU in the Extension Plant Sciences Department. And so a little bit more about him can be found in your program. But Dr. Lujan is the plant diagnostician for the NMSU Plant Diagnostic Clinic. He received his BS and master's in agricultural biology with a minor in molecular biology and PhD in plant and environmental sciences with an emphasis in plant physiology. Excuse me.
Sorry.
I said it wrong. Plant pathology. Thank you. So he was a research technician in the plant pathology and mycology program at NMSU, where he gained valuable experience working with local growers, helping them to manage their disease problems. And he also fields all of the samples that get sent into the diagnostic clinic from around the state. So with that, thanks so much, Dr. Lujan.
Thank you. Thank you, Dr. Thompson. Can everybody hear me pretty good? Good. Okay. So I'm going to be talking about the top 10 tree diseases and disorders that is found in New Mexico. As Dr. Thompson mentioned, I do run the Plant Diagnostic Clinic, and so it's a lot focused on New Mexico issues, but we do accept, plants from around the contiguous 48 states. So I'll talk a little bit more about that later on. Let's get started. So it is not moving forward. Am I supposed to do something else? It's moving forward here. Yeah. Nice. Okay. So I tend to give a little bit of a talk outline. The beginning is going to be a general plant pathology, just to get everybody up to speed, with some definitions and things like that. And then I'm going to be talking about the Plant Diagnostic Clinic, and you can use that as a resource, if you're in New Mexico or even around the region. So just get to know us a little bit. And then we'll go into the top 10. I don't know the comedian that does the top 10, but this is not that. We'll discuss plant pathology and plant diseases. So not so much fun, but I think it is. All right, so the beginning, general tree problems. There are many different living, which we term biotic, and non-living, which is abiotic entities that can have a negative effect on your trees. Trees that are under stress are much more susceptible to diseases than trees that are, healthy and growing strong. I tend to, compare it to, a human being, right? So if we're getting our exercise, we're eating right, we're getting our vitamins in, we tend to fend off some of these, general common cold issues, versus those that are more sedentary, their lifestyles aren't that great. They tend to have a little bit more of a disease than they can get more diseases than healthier individuals. And then successful disease management, that really depends on first the accurate identification of the causal agent and the integrated use of available control methods. And so I underline the accurate identification of the causal agent, because a number of times we go out to field visits and, people say, I've sprayed this fungicide, when in fact it's not really a fungal issue. So it's not really doing anything besides maybe out of pocket and some environmental issues. So that accurate identification of, what is actually going on with the tree causing those issues, that is pivotal and very important. So now moving on to a little bit more of definitions between a plant disorder and a plant disease. So I listed on the title, the top 10 diseases and disorders. A plant disorder really is just any abnormal growth or development in the plant. And that can be both abiotic, environmental, cultural issues, but it can also be a plant disease, right? Because that's just any abnormal growth or development within the plant or tree. Now, plant disease is a little bit more specific. A plant disease, again, it has that abnormal growth or development in the plant, but this is specifically caused by an infectious microorganism, or what we call a pathogen. All right, so I've been talking a lot about causal agents and biotic and abiotic. What are those? For those that are not familiar with those terms, just a little bit more of an example of what those are. And so some biotic factors or living organisms that can be, fungus, that can be bacteria, viruses, nematodes, those microscopic worms that are found in the soil, parasitic plants or parasitic higher plants like mistletoe that we see, or after a rain, you see the stringy yellow spaghetti that's on the ground, which we call dodder. That's also a parasitic plant. Insects, which Dr. King will be talking a lot about, and then weeds, which Dr. Beck will be talking about. Those can, the insects can physically damage plants, and then as well, they can also vector some of these viral diseases, and affect your trees that way. And weeds also have been known, we've seen that they also harbor a lot of diseases as well, so they can spread that way if you keep weeds around. They can also help insects to overwinter in those weeds. So sort of manage your weeds, and you'll hear all about that from Dr. Beck. Now, you can see these diseases or biotic factors, they typically spread from plant to plant. You see it on more of a, patches or a smaller area, versus those abiotic factors or non-living. Those can be temperature extremes here in New Mexico, right? Very hot in the summer. Sometimes we get really cold in the winter. So those also play a role in, causing some damage. Moisture extremes, again, not enough water, or on the opposite end, too much water with our downpours. Soil problems. I know in Las Cruces, we don't have the greatest of soils, so that's always an issue. Soil health is sort of a big topic right now. So a lot of soil issues or soil problems can sort of predispose some of these trees to diseases. And then you get nutrient deficiencies and toxicities, as well as wind, right? If we don't plant deep enough, or if we plant too shallow, that wind will just immediately just knock it down. Pesticide toxicity is always an issue. And then just improper cultural practices. I'll be discussing this a little bit more on a further slide. But these typically do not spread, from one tree to the next. Why is it going forward? You get more of a larger area, sort of a distribution or a uniform pattern. So if you can look aerially, you may see more of a pattern that way. That kind of leads us to think more of abiotic issues versus biotic. All right, so a little bit more of actually identification of the causal agents. So this is when a sample comes into my lab. We try to tease apart if it's either abiotic or environmental versus biotic or an actual pathogen that we need to explore a little bit more. So these are all generalities. Of course, there's always, those, unique cases, but typically more than one plant species is affected. You see a tree, it's affected. You see, grass around it. You see other plants that are around that area, all showing the, same symptoms. That's typically more of an environmental issue. From one day to the next, your tree's dead, your tree looks really awful, or your plants look really awful. The symptoms develop really very suddenly. That leads us more into thinking that is more of an environmental issue. And then the symptoms, as I mentioned before, those typically do not spread. They're pretty localized. And then the distribution is typically uniform. Now, when we think of a pathogen, pathogens are fairly plant specific or even general specific. Typically, the symptoms develop very slowly, right? So it'd be a horrible pathogen if it starts to, kill the tree within a day or two. It would have to find a new host. So that takes a lot of energy, effort to do so. So typically, it's sort of, gets in there in the vascular system, starts utilizing those nutrients, and then starts to grow that way. And then it starts to move on after that. And then symptoms spread from plant to plant. So you'll see one tree, maybe that's affected either through the roots. And then you start to see another tree that looks very similar, and it's also starting to show some symptoms. And then the distribution, as I mentioned before, is typically scattered, clustered, or sporadic. Now, every plant pathologist loves to talk about the plant disease triangle, right? So in order to get disease, you need to actually have 3 different things happen all together. You need to have an actual virulent pathogen. So a pathogen that's either in the soil, in the air, but it's able to cause disease. Again, as I mentioned before, fungi, bacteria, viruses, nematodes, and phytoplasmas are within that realm. Next, you need to have an actual susceptible host. So if you have a pathogen there and you have a host that's tolerant or resistant to that actual pathogen, then you're not going to have disease occur. Now, if you do have both of those things, you have a pathogen, you have a susceptible host. Now you need a favorable environment in order for a disease to occur, right? So typically we either in the dead of summer or the dead of winter, we typically don't really see too many disease issues occurring. And that's because the fungus, it's either too cold or too hot for it to even grow. It prefers pretty almost like us, 75 is really good. A little bit of humidity, but not so much, but also not very dry. So we typically like fungi, bacteria, they typically like that favorable environment. And so after a good rainfall, a few days after, a week after, you start to see some things develop. And so that's when a disease occurs. It's when you have all 3 of these things working together, then you can develop the plant can develop a disease, not yourselves. So with this in mind, we can use this to our advantage here. We can try to alter some of these things, right? So maybe the pathogen, maybe it's in the soil. We can do a few things that way, but maybe if you know the history of that soil, maybe plant a more susceptible type of tree, or not a susceptible, more there we go, more a resistant type of tree for that specific pathogen. Or we can alter, that microclimate, right? So alter that, maybe selective pruning to allow that, the wind to go through, kind of lower that humidity that way. So just think about that when you're thinking about diseases, those three types of things. Now moving on, a little bit more definitions, symptoms. So those are the actual responses of the plant, either to an attack by an actual pathogen or to adverse conditions, caused by the environmental factor. Examples of that: yellowing or chlorosis, wilting, leaf spots, dead or rotting parts, or stunting. So those are all examples of symptoms. Now, there's so many times that I've been, sent a picture with a leaf spot and, saying, what is this? I don't know, because there are a number of things that can be going on. We can't just identify the problem just by looking at the symptoms. If you see it often enough, you have that experience, maybe you can make a little bit more of an educated guess. But really, just by, a fuzzy picture normally, you can't really identify that type of problem. And so a little bit of an example here, just some general spots, general symptoms develop to show us, that there is a problem. And so these are all leaf spots. One is caused by a fungal issue. The second, any guesses? Fungal, bacterial, environmental? Shout it out. Anyone? Bacterial? Okay. Well, it's a fungal. So, that's how it goes, right? If you get a picture like that. The next one, bacterial. They all pretty much look the same, right? You have a little bit of necrosis on the inner part. You get a yellowing halo around the edge. And then finally, this one's a little bit more of a giveaway because of just the type, that type of symptom. It's more of a viral issue. So this is just an example of the types of things that we get. And just because we see spots doesn't necessarily mean that, this is one specific fungus or one specific disease. Now moving on to signs. So signs are the actual visual presence of the pathogen on the plant. And so examples of this are, rust, pycnidia, which are these little, on the second page or second picture here, these little black things here are full of spores. So those are just the housing structures for spores. And once that environmental condition occurs, you get a little bit of moisture, you get some humidity, some wind, it starts to shoot up those spores and start infecting other things. You can get some bacterial ooze here. And then this is the actual fungus, the powdery mildew that's on these cherries there. So, let's see. So if you do see signs, that can sort of lead us to more. We know that it's a rust, or we know it's powdery mildew. So that gives us a little bit of more versus the symptoms themselves. Now, disease management. The key to disease management, right, is prevention. So well-managed trees equals healthy trees. The most effective management programs incorporate all strategies to reduce disease severity. And so we term that in the plant pathology world as integrated disease management. Now there's about 4 of them. There's physical, examples of that is pruning, cultural practices such as irrigation, biologicals, so using maybe some mycorrhizae, some beneficial fungi like trichodermas, bacillus, really good health bacteria. You can incorporate those into the soil. And then, of course, there's chemical. With chemical, I didn't put it in there, but I'm sure you'll be hearing it often. You need to read the label. The label is a law, so you need to, if you are applying that, you need to make sure that you're applying it correctly according to the label. Now, accurate, again, I'm going to say this over and over again, accurate identification of the disease pathogen or disorder is really that first key and essential for successful management. Now, for us in the lab, sometimes, to add more trouble versus, you get a sample in, it's pretty much dead. You start to see other things growing. Well, there's organisms associated with diseased trees or diseased plants in general. So you get that primary organism. So that's the actual pathogen that's responsible for or directly responsible for that disease occurring. If we don't catch it in time, if you're not, scouting well, if you, take a sample, but you leave it in the truck for a few days, you start to get other things growing, right? So these secondary organisms, those are the ones that take advantage of a weakened plant tissue. And they grow really quickly. And so in the lab, when we're trying to, tease apart the secondary versus a primary, oftentimes that secondary just completely outgrows that primary. So as soon as you start to see something and you suspect that, you want to get an identification through the lab, you want to sample it, send it in right away, work with your extension agents, but I'll talk about that a little bit later. Now, these weak pathogens, these are secondary organisms, but what we've seen recently in the past, 5-6 years is we're getting a lot more of these secondary organisms or weak pathogens becoming actual primary pathogens. And that's what we think is due to the increasing climate stresses. So again, as I mentioned, those abiotic factors oftentimes predispose plants or trees to infection. And so these weak pathogens are becoming more of a primary problem. So, that's kind of concerning as well. And then finally, we have our saprophytes. And these are just non-pathogenic microorganisms. They come in last, once the plant tissue is pretty much dead, and they feed off of that decaying organic matter. And so these saprophytes outgrow these secondary organisms, which outgrow the primary. So again, if you want to send a sample in, try to get that primary organism or send it off to us as soon as possible. Now, test, don't guess. A little bit about the Plant Diagnostic Clinic and who we are, right? So diagnosis or trying to find out what's causing the problem is really a team effort. Starts with the grower, producer, or those people that are, those boots on the ground people first noticing that there is an issue with their trees. Then we have, submitting agents, whether that's your extension specialist, your county extension agent, or even if you want to just directly, working with a company, you can directly send it to the lab. And then finally, everything sort of, if you want to send it to the plant clinic, it ends up at the diagnostic lab. So a little bit about who we are. You'll see a few of these people in later talks. And so the personnel, the director or the weed specialist is Dr. Leslie Beck, who's here. I'm the diagnostician. There's associated personnel. So there's, people that specialize in entomology, like Dr. Joni King, who's also here. We have horticulture specialists like Dr. Marissa Thompson. And then we do have a nematologist on board with the clinic. So if you suspect that there are some nematode issues, then you can send those to Jackie Beecham. Some of the services that we do provide there, we are an integrated plant diagnostic clinic. What that means is that we can handle diseases. We try to figure out abiotic issues. If there's not a disease, arthropod identification and damage, and as well as plant and weed identification. So we do have a USDA permit that allows us to receive all plant samples from the 48 contiguous states. Hawaii and Alaska, we don't want to bring anything from there into the contiguous states and then cause a whole issue. All right, so now moving on, I am sort of the bearer of unfortunate news. Plant diseases are very difficult to cure. That's especially true when the diseases are well established within and throughout the tree. So those systemic diseases goes from the roots up to the vascular system and out. Oftentimes we don't really notice, right, until we start to see little things, little clues that shows us that there's some issues. When those symptoms are clearly noticeable, oftentimes it's too late to effectively treat. So really, oftentimes it's just more of management strategies aimed to reduce stress, prolong life, and also try to just prevent death as much as we can. Now, some just general treatment management considerations. Again, just critical evaluation of each unique situation is necessary. There's not really a one-size-fits-all solution, right? So if we get a plant or a branch in that has some sort of issue, we can't really go through and say, well, we would do this. We need to, look at the surroundings, look at what's growing around, and then we develop sort of a better management plan that way. So again, things to consider: likelihood of treatment success, the hazardous nature of the tree. So if it's completely dead and it's about to fall off or, fall over, then we want to reduce that risk. The cost of the treatment, whether you have enough labor, the equipment's there. The tree value, sometimes sentimental value is extremely important with trees. And so we just don't go in and say, well, I would just chainsaw that out and get rid of it. So we need to understand, some of these management considerations. And then just the impact on the environment and, animals as well. All right, so now moving on to the top 10 trees submitted to the NMSU Plant Diagnostic Clinic. This is from when I first started, so from 2018 to current. Here's the top 10. We put them from genus, common name, as well as a % of total tree samples submitted. So you can see at the top, the most common plant sample or tree sample that's been submitted are pine, juniper, pear, ash, and then it starts to sort of tail off. Now the top 10 tree disease and disorders, I was going to do like from 10 to 1, but I think it's a little bit better here in the table format in case people want to take pictures and they can kind of see, you know, a little bit better here. From 2018 to current, we received about 825 tree samples to the clinic. And so 61% of those trees, so more than half, are, you know, actual abiotic issues. Sort of makes sense here, right, in our climate. Those are cultural, environmental issues, common hosts, everything. 10% is Phomopsis blight or canker. And so I'm going to go through each of these a little bit more in detail, but just kind of wanted to give you all a general picture or an outline here. For Phomopsis, we typically see it on pine and junipers, but it can happen on spruce, mulberry, and poplars. Rhizosphere and needle cast is another one at 8%. Then it starts to tail off. So fire blight is another bacterial disease. Cytospora canker is an interesting one, and one that I mentioned being, you know, a secondary organism type of problem, but it's sort of kind of creeping up into more of a primary organism issue since I've been here. And then iron chlorosis, bacterial canker, Phytotoxicum root rot. So there are folks here from the southern part of the state. A few of us. Not so many. Okay, so, well, that one we can maybe skip depending on time. That's more of a pathogen that's restricted to the southern half of New Mexico. So I think the furthest is Sierra County down. So anything above that, not so much. All right, so let's talk about these things in a little bit more detail. The first, again, abiotic stresses. So if you've received, you know, a report from us, sometimes we say cultural, environmental issues are a factor. And so what that is, maybe irrigation or drought, planting area, planting depth, just inappropriate growing trees for your growing zones, and then these microclimates. And so this is, I can safely, I think, say this is in Las Cruces here. This is in summer 2021. These are just what it looks like maybe in the winter, maybe. But these are all ash trees and they're not looking too happy. And so that's more after some, you know, we took samples, we try to figure if it was a problem or not, or a disease. It wasn't, and so that's more of a cultural environmental issue. The urban growing environment is, you know, pretty stressful for trees. Again, they can predispose trees to that secondary pathogen infection. So just a little bit more pictures around, you know, some more healthy, but you do see some turf around here. These maybe not so healthy, and you can see some dieback starting to occur, some chlorosis on the trees as well. Again, the abiotic stress, and think about that a plant disease. So with this one, we don't have an actual pathogen, but we do have a host and favorable environment, right? As I mentioned, those are two of the three legs, you know, that can cause plant disease. So you have two of them. All we need now is something to blow in, and you'll get that 3, and then you'll start to develop plant disease. So again, just predisposes trees to infection. We have an enormous amount of reference material with our NMSU extension guides on, you know, all horticulture, abiotic stresses, everything like that. You can find that at pubs.nmsu.edu. All right, so now moving on to the second one, that's Phomopsis blight and canker. So it's caused by the pathogen Phomopsis. The species are pretty host specific, so depending on the host that's submitted, we can get a Phomopsis, but with the type of species. Common hosts that we find it on are cottonwoods, junipers, and cedars, and small fruits. So those spores are spread by wind, rain, insects, natural wounds, and pruning. But, you know, typically Typically, they're housed in this pycnidia structure, but when moisture occurs, then they start to release those spores. The optimum temperature for infection is about 75 degrees Fahrenheit. And then disease development, you start to see around 90 degrees. It's a serious problem in nurseries as it can spread quite easily through pruning or wounds. Really, landscape trees, they're rarely killed, but they can become sort of an unsightly and unaesthetic with numerous dead branches occurring. Some of the symptoms that we do see for Phomopsis infection are dieback and blight. Cankers develop around the larger stems, and then the smaller stems oftentimes are girdled, so they're the vascular system is really constricted, not allowing any nutrients or water to go up to the top. So anywhere that vascular system is restricted, then you're going to see death occurring. Now, signs, again, if we look closely, sometimes we can see those black fruiting bodies, and when in the lab, we can tell what that is fairly quickly. With all of these, I'm going to provide some integrated disease management options. This is a weak pathogen, so prevention is pretty much where you start. Avoid unnecessary injuries. You want to reduce stress, so proper water, fertilizer if needed. You want to avoid wetting the foliage because then, as I mentioned before, you need that moisture and that wind to start spreading the spores. You want to prune out infected twigs and branches if you do see any, but you also want to make sure that you clean your tools when doing so, because that can be a way to spread those diseases. And you never want to prune when the branches are wet. Third is Rhizosphere needle cast. Several fungal pathogens cause evergreen needle diseases. They can affect the foliage, they can affect the buds, as well as the shoots. Major hosts that we find this in are spruce, especially blue spruce and pines. And so fungi sporulates in the late winter and early spring. And then the spores, again, as before, they're spread by wind and rain. The symptoms do not really develop until late summer or fall. Sometimes you see an off color sort of starting to turn yellow and then eventually become necrotic and brown. And then those dead needles may soon drop after symptom development. So that needle discoloration, oftentimes it's from yellow, so you get a little bit of a reddish purple tinge, and then you turn brown. Sometimes it can be oftentimes it can be confused with winter injury because they look similar. But with this one, it usually starts developing from the bottom up to the top and from the inside out. So that's sort of a better way to look at it. And then the signs there, if we do see some needles within the sample, you can look to see if there's little black pycnidia. We smash that under the microscope and you can see the actual spores and mycelium that way. For disease management, again, I'm going to be probably saying this every slide: provide accurate water, reduce your stress, avoid wetting the needles because that can spread. You want to promote good air circulation around the trees. That microclimate, you can develop a pretty humid area around there, and that's just perfect conditions for that fungi. If caught early enough, fungicide treatments can be very effective. So a Bordeaux mixture, and then as well as some chlorothalonil fungicides as well. Read the label though. The next one, fire blight. This is caused by a bacteria called Erwinia amblivora. The hosts are trees in the Rosaceae family, so apples, cherries, peaches. It overwinters in previous years' cankers and healthy buds. So in this picture here with these red boxes, you could see the actual canker that's developed, and so that's where the bacteria is presently located. Until you get that environmental condition for it to spread, and then it grows like wildfire and starts, like fire blight. The optimal conditions, about 75 to 85 degrees, greater than 55 degrees at night with some intermittent rain. It is spread by contaminated pruning tools, insects, and bees. You know, some of the symptoms are infected twigs, again, those water soaked and dark and sunken lesions. And then at the terminal ends, you can see it looks like, you know, it's pretty very descriptive, the name, where it looks like a part of it's been set on fire. It often, you know, bends. And so what we call a shepherd's crook, if you see that kind of sticking out, it looks bent, it looks burned, you see some of these lesions, you can sort of call that it's a fire blight issue. For management strategies with this one, start with disease-free planting material. So you want to carefully inspect new material. If you do see some of these cankers here, you want to prune out at least 6 inches below the margin of that canker. So if you see it here, you want to move 6 down and start pruning that way before it starts spreading. Again, you can spread it through infected tools, so be sure to clean your tools afterwards or moving from orchard to orchard. Protective sprays are also available, copper fungicides or antibiotics. Typically, that's only cost effective for more commercial operations. The next one is Cytospora canker. This is caused by several fungal pathogens. Host are apple, ash, cottonwood. The fungi attacks trees that are, you know, or parts of the trees that are in a weak or stress condition. As I mentioned before, this is more of a weak pathogen, but we're starting to see more of it. Again, emerging issue with climate change. And so if we have a sample that comes in, you can see where the healthy tissue is and start of that fungi attacking that vascular system. Some of the symptoms that can occur with trees that have Cytospora canker, it can vary by host, but typically you do see a branch dieback. You start to see cankers develop. The inner bark can turn reddish to brown to black, as I mentioned here. Now, signs of infection, it's not very clear, but you do see some of these orange or red tendrils. So this was at actually my parents' place on an ash tree, and we started to see these orange tendrils. I got really excited as being the plant pathologist. My parents didn't like it because at that point, you know, the pathogen was throughout the tree, throughout the vascular system, and it was almost time for it to be gone. The ooze, you get liquid ooze on aspen and gummy ooze on peach and cherry. So for Cytospora canker, again, purchase healthy nursery stock. You want to inspect your new material. Because this is, you know, a disease that occurs on a weakened host, you want to prevent stress through proper irrigation as well as cultural practices. And there are a lot of references to, you know, proper selecting ornamental trees, shade trees for New Mexico, fruits and nuts for New Mexico orchards. So we have a lot of extension material available through our websites, NMSU websites. And then you just want to avoid physical wounds because those are just entry points for you know, anything to get in there. And then you can apply labeled fungicides as wound dressings as well. So iron chlorosis, I'm not going to spend too much time on this, but it is one of the most common nutrient disorders found here in New Mexico, or at least that are submitted through the clinic. And so iron is an essential element for normal growth and development, but typically you can start to see symptoms of iron chlorosis beginning in the spring when the trees are actually leafing out. And so symptoms include interveinal chlorosis. You get those yellow, bright yellow leaves with those dark green veins. Management, applying the correct type of iron at the correct time is especially useful in New Mexico, where we have very calcareous soils. That calcium just kind of binds that iron and not allow it to be available for the tree. So we also have an extension guide specifically talking about iron chlorosis and the correct type of iron to be purchased. All right, so the next one is bacterial canker. Other names are gummosis, blossom blast, spur, and twig blight. It's caused by a bacterial pathogen called Pseudomonas syringae. Its primary hosts are, the stone fruit trees, but it does have a wide host range. It overwinters in infected tissue. Sometimes you'll see symptoms, sometimes you won't. So this one's kind of a tricky one. It can also enter through injured areas, whether it's pruning, freeze, sunburn, or hail. High humidity, like most bacterial issues, is needed. And then you get rainfall and cooler temperatures that favors infection and disease development. Now, some of the symptoms of bacterial canker, as the name implies, you get cankers with or without gumming. You get a thinning of the tree canopy. You get some leaf drop and dieback. You get blossom blight, and then sometimes you can also develop, the tree can also develop fruit spots as well. Now, management strategies for this, again, protection from injuries, reducing stress, sound like a broken record, but those are very important, keeping your trees healthy. There's a chemical control, so copper sprays can be applied both in the fall and the spring. Those need to be applied before flowering. And then I think I can sort of just run through this really quickly because this is more of a southern issue. But Phytotoxicum root rot, also known as cotton root rot or Texas root rot. If you're in Texas, if I was in Texas giving this, I would never say that, but because they do get upset. But this was first found there, so that's what it is. The pathogen is actually Phytotrichopsis omnivora, and omnivora means it eats everything. So the host range, has over 2,300 different plant species that can be affected by this. From pine, fruit trees, ash, we see a lot of it in Doñana County and pecan. But luckily for you all up north, the fungus is pretty limited geographically to parts of Arizona, southern New Mexico, a good part of Texas, and a lot of Mexico. It's only found at elevations below 5,000 feet. And then even within that, you can see just a picture of a field here using, I guess, a drone. You can see it's very just patchy, and from one area, you could just see complete death. And then just a little bit more, everything's fine. So it's also a pretty tricky disease. Found in soils and low organic matter and high alkalinity, so perfect in southern New Mexico. The spread of the fungus, luckily, is very limited. So it doesn't really spread by spores. It only spreads by root grafts. So an infected root needs to be touching another root. But unfortunately, it survives very deep in the soil. So you can't, trying to manage the soil, this thing can survive over 12 feet deep. And then it also makes sclerotia. So sclerotia are, just think of it like a ball of yarn. The mycelium or the fungus actually wraps itself around very tightly, becomes melanized, so it becomes black to prevent it from UV degradation and drying out. And then it can survive in that soil for 20 years, down deep, until you get those perfect conditions. It unwinds and starts infecting that way. Typically, the disease appears in summer or the fall, 85 or above, with some moisture. And then this one is a pretty aggressive pathogen. From as little as 2 weeks, you get a healthy tree, you start to get infection, and then within 2 weeks, you get just a completely fried tree. Signs, after a good rain, you may see some whitish tan spore mats develop around the base of the tree. And then for root samples, you can get, let's see here, fungal strands on the roots. So if I look under a microscope or with a hand lens, oftentimes you can see it, this fungus kind of growing alongside that root right there. Management is extremely difficult, if not impossible. So this one's a very, just a horrible disease. People have been trying to change the soil environment, but that you would have to constantly be doing that, applying a lot of organic matter, applying ammonium sulfate and sulfur to reduce the pH. And then if you don't do that one season, it goes back up. And so it's a constant struggle. Other scientists have been looking at green manure cover crops as well to stimulate the rooting of the trees to withstand some of that disease pressure, and then incorporating that cover crop to stimulate some of that soil beneficial microflora as well. Slime flux is next, or bacterial wetwood. Several species of bacteria can cause slime flux. If you want a really good, if you want to take a tour of NMSU, you see a lot of slime flux. Yeah, so the hosts are fast-growing trees. Those are especially susceptible. Willow, elms, cottonwoods. The bacteria enters through physical wounds or through natural growth cracks. And once inside, it sort of just raises that internal tree gas pressure until it's forced out, and you start to see this occurring. You see these little holes. The slime produced is pretty toxic, it's very acidic, so you start to see that acid washing there. Some of the symptoms, you get that ooze flowing down the trunks or branches. You get more of like a moist bark there. That's what is called bacterial wetwood. And then it dries to that whitish color. Insects tend to be attracted to that ooze. It's because they like it, I guess. It sometimes stinks. So once infected, there's not really a cure. So again, prevention, try to avoid wounding the tree around the base where that bacterial is present in the soil. Proper irrigation to reduce the stress. And sometimes that if you do see slime, you can wash it off of the trunk and limbs, sort of reduces the toxic effect of that bacteria just by dilution. And then finally, number 10 is bacterial leaf scorch. The pathogen is a bacteria, Xylella fastidiosa. It is a plant pathogen, but it is also found in a lot of trees as a non-pathogenic endophyte. So it's there, but it's not really causing issues. It's a disease in about 10% of trees, and the other 90% maybe an endophyte. Hosts, oak, Mexican alder, desert willow, catalpa. The xylem, it's a xylem-limited bacteria, so it resides and multiplies within the xylem. It looks like drought symptoms or a nutrient deficiency, where you get leaf scorch, yellowing, reduced growth, and dieback. And then this bacteria can be transmitted through xylem-feeding insects such as sharpshooters, spittlebugs, and leafhoppers, as well as through grafting. As I mentioned, it has been proven to be a common organism in nursery stock, both symptomatic and asymptomatic, so more of that endophyte type of thing. Unfortunately for me, my job is very difficult with Xylella because detection is very difficult. That bacteria, because that fastidiosa is attached to that name, means it doesn't like to be outside of its host. So it doesn't really grow well in culture. So that kind of limits what we can do. There are DNA tests that are used to detect the pathogen, both ELISA and PCR. It is time consuming, a little bit expensive. And then some of the metabolites that are naturally produced in trees can sort of inhibit detection by these methods. There's a new piece of equipment that we got a couple of years ago. It's called isothermal amplification. So it's helped us a lot with detection of Xylella. It is very quick. Within 20 minutes, we can figure out if there's Xylella or not. It's very sensitive, but it's also pretty expensive. And again, bacteria are not evenly distributed throughout the tree. So sometimes you see a really symptomatic branch, you send that in, it's negative. You see what we think is a healthier branch, it's positive. So sometimes we need a fair amount of sample for this one if we do suspect that it's bacterial leaf scorch. All right, so I am rounding the end, but I just want to be sure, not everything you plant is going to die because of disease, right? I want to make sure that, disease only occurs if there's a pathogen, there's a susceptible host, and there's a favorable environment. So just be thinking about that when you think this is dying of disease and maybe what we can do about it. Again, key to disease management, I think I've said this every slide, prevention. Well-managed trees equals healthy trees. And so use those integrated disease management program. But first, again, accurate identification of the disease and pathogen or disorder is essential for successful management. And so that's why we're here at the NMSU Plant Diagnostic Clinic. So if you have any issues, just contact us and we'll be more than happy to help you. And so if you have any questions about the presentation, tree problems, or you don't know where to get started with sampling, what should I sample, contact me. I'm happy to talk to you. Email is usually preferred, so I can give out a nice list of what to do instead of over the phone. Then sometimes we miss something. So email is usually good. I'm usually also running around between greenhouses, fields, office, and labs. And here are some websites that you can visit. They have sampling sheets, as well as all the extension publications that we have at NMSU. And with that, I did put it in there. There's the code, I guess, for those that need it. Is it on? Okay, it is.
Have you been monitoring things like Texas root rot for possible changing in scope of area because of climate change, moving north, or any other diseases because of climate change, and how that may affect where things are found at this point?
Yeah, so that's a great question, and climate does play a huge role in where we find diseases and disorders, and so that is good. Texas or cotton root rot is more of an elevation issue. So, I mean, it could be expanding, but we still haven't seen it past that Sierra County. But we're always on the lookout for it. But yeah, we're always scouting and making sure that we don't see new things because of the changing climate.
We're working with Pinon, trying to save that around homes and stuff. And I've heard the Colorado State Forester said that perhaps the Pinon might be listed as a T&E species. And do you know anything about that with Ips beetles or any information on those pathogens?
That we do get a lot of Pinon issues, and there are a lot of Ips beetle issues, and so that may be more of you may want to save that one in your back pocket for the entomologist that may be teed up for that question soon. Tomorrow she'll be speaking about that, but we do have a lot of Pinon canker that we see. But that one's kind of the one that we do see, and needle scale as well.
Thank you. I have two questions from the streamers. One, I also have a question myself. Oh, three questions. One, John White asks, are the blackened wounds on mesquite trees from slime flux?
It could be. It's tough to just generalize a blackening on anything really, but it could be because of slime flux, but it could be just other things as
Yeah,
well.
it'd be like a city mold.
Yeah.
And
city
so
mold was good.
I think, John, you should send a sample to the diagnostic clinic.
Yes, John, you know how to
Um,
do that.
And then Paul Brian Jones is also online from Taos, and he asks, how long do samples take?
That sort of depends on when we receive the sample first, and then some pathogens take a while to grow out. And then, so between, 3 days to a week and a half, we try to do our best to communicate with those people that do submit a sample, if it's going to take a little bit longer than a week, just to be sure, we are working on it. We do receive a lot of samples, especially in the spring and summer. And so we're constantly working on it, but there are pathogens that take a fair amount of time to get out of its host and then grow, and then we can identify it that way.
Thank you. And then George asks, is bacterial leaf scorch host specific?
No, it's fairly general. It has a lot of hosts actually, but again, as I mentioned, it can be found as an endophyte, so it can just be there and not really causing any issues. So we can detect it, or we cannot. So it's all, symptoms versus an endophyte. So.
Thank you.
Yeah.
Okay. And a question I have is, can you speak to mistletoe range changes, also climate crisis related?
Yeah, so people have been reaching out a lot. I wanted to put like an 11 maybe. That mistletoe, like on cottonwoods, we see those a lot, and those are starting to, if there's a lot of mistletoe on a tree, it's of course it's a parasitic plant, so it's getting nutrients and it's starting to, inhibit, I guess, the good growth of a tree. So that can be an issue as well. And we are just seeing a lot more of mistletoe popping up around.
Thank you.
Mhm.
Okay.
There might be.
Thank you. Yes. Aaron. Yeah.
Question. I've been curious about tree growth regulator, especially it's like got fungistatic properties and possibly bolsters the tree's ability to withstand drought and bolster its self-defense. And curious if you know anything about that or have seen any research, specifically thinking of pinon needle scale and maybe like the cytospora and things like that. If just curious.
Yeah, I think that I think maybe you want to talk to the horticulturist in the room may want to talk about that.
Thank you. I don't know specifically for that. I'd like to look into that more. Maybe there's room for research in New Mexico on that. I studied plant growth regulators and their effects on flowering for my PhD, and I vowed to never work with them again because it was so complicated. And there's this thing called hormone crosstalk, right? So it's like it's not necessarily just the presence of one or other of these natural hormones. It's also their ratios in relation to each other. So I don't have an answer except that it's probably complicated. Thank you.
All right, thank you.
Okay, with that, thanks so much, Dr. Lujan. I do want to give you one of our tree cookies. So one of our board member, George Duda, who is also wearing the chainsaw hat, makes these tree cookies each year for us to give away, and they're beautiful. Thank you so much for coming to present.